So neonatal sepsis happens when babies get an infection in their blood. It can be very risky. It can cause severe illness and even death, especially if they are born too early or have a weak immune system. Traditionally, diagnosing sepsis is done through blood tests, which is time-consuming. So this will delay the timely intervention of medical professionals. To tackle this, doctors often initiate empiric antibiotic treatment for newborns while waiting for the blood results. So this early administration can affect negatively the development of the immune system of the babies. It will result in long admissions in the intensive care units and contribute to increased in healthcare costs, posing emotional and financial challenges on families. However, I'm here to talk about a new approach that goes beyond traditional methods and combine artificial intelligence with nanotechnology. Another interesting aspect of this project is microRNA. So microRNAs are tiny messengers that can influence how our body acts in case of an infection or disease. So what's remarkable about microRNA biomarkers is their discrimination capability. Meaning they can distinguish between different stages of infection with high accuracy. And in addition to this, they can be analyzed in small blood volumes, which is important for neonates and newborns. And later, this usage of microRNA is important because they are stable under different conditions. These attributes altogether make microRNA a promising avenue for more accurate and less invasive monitoring and diagnosis for neonatal sepsis. So what are our exact aims? First, we aim to identify and validate microRNA that are predictive for neonatal sepsis in biofluids using two different cohorts for discovery and for validation. Second, we aim to design a specific sensor with specific chemistry to detect the panel of microRNA we already have identified. Third, we aim to collect biological data from different biobanks around the globe in order to collect important information about patients. And with that, we can identify early signs of infection and by training a machine learning model. Lastly, we aim to integrate this pre-trained machine learning model with our QCM sensor, with the data of the QCM sensor for rapid detection of sepsis in neonates. In the upcoming slides, I will break down each aim for you. So to identify diagnostic biomarkers, we will enroll patients from 3 different hospitals in Lebanon, including ABMC. Following enrollment, we will need to collect blood, urine, saliva specimens from babies. After serum isolation, our goal is to identify microRNA or different microRNA that are predictive for neonatal sepsis. And for example, we have microRNA 14.6a, microRNA 15a, and 16 who are upregulated in serum samples, while microRNA 26a is downregulated. So furthermore, the identified panel will be validated in a separate cohort for more reliable results. Moving forward, our aim is to develop a diagnostic tool using quartz crystal microbalance biosensor. It consists of top and bottom electrodes coated with gold and a quartz crystal plate. So the aim of the sensor is to detect microRNA binding events on its surface, so it might increase the mass change on the surface, and we can correlate this and convert them into signals. At this stage, we focus on developing a biosensor that is user-friendly, convenient, and low-cost, for quick and accurate detection of sepsis. It's worth noting that quartz crystal biosensors can detect variations of mass down to 0.1 nanograms. In this slide, we illustrate the process of attaching a microRNA after cleaning and activating the surface of the sensor. We first introduce the probe microRNA, which is labeled as P1, to the sensor's surface. After washing and removing P1 on the surface of the sensor, delta F1, a drop of a frequency occurs, highlighting that there is a mass change on the surface of the sensor, meaning that there is P1 attached to the surface of the sensor. After that, we need to introduce the target of interest we're looking for in the samples of patients. So P1 and target get attached to each other due to complementarity. So another drop of frequency occurs, which is delta F2, meaning that there is an increase of mass on the sensor surface. Later, to increase and to enhance sensitivity of the sensor, we will introduce P2, which can be binded to another part of the target to enhance signal detection. In another context, we will deploy machine learning model to analyze complex patterns in biological data for the detection of sepsis. To do so, we have to collect data from different biobanks around the globe. These sources will help us gathering information about patients such as age, weight, gender, clinical data, for example, vital signs, and more importantly, microRNA expression levels that can be counted using microRNA sequencing. Altogether, are necessary to train a machine learning model for sepsis detection. This data set will serve for training, testing, and validating the employed machine learning model. Lastly, we will integrate real-time information provided from the biosensor along with patient information and vital signs of the patient in question. This integration will enable us to predict the presence or absence of sepsis with high accuracy. Therefore, a final output of this project will be a prototype microRNA-based point-of-care diagnostic tool that will help decrease the morbidity associated with sepsis and lowering the overall cost of care. Of course, this will assist doctors in clinical decision, improve treatment strategies, and optimizing patient outcomes. In conclusion, this approach represents a step forward in neonatal care. Through the integration of machine learning with advanced nanobiosensor, we can improve the quality of life of newborns. In addition to this, we can use tiny samples to detect sepsis with high sensitivity and specificity. So this empowers healthcare providers to give a timely intervention and which is prevent the progression of the infection in newborns and improving their quality of life. Moreover, our approach has the potential to reduce healthcare costs, especially for parents, and importantly, these benefits can be achieved with high accuracy and efficiency. Thank you.
Get from what you presented that this is still at the very beginning, right?
Yes.
You're still initiating that. So can you comment on regarding the planned QCM studies? So you know QCM measures mass but also measures dissipation if we're using a device that we have currently here.
Yes.
How can you actually use dissipation in your approach, or is that relevant at all?
Actually, we're working with samples in wet conditions. So there is actually dissipation in it. So we will taking into consideration this dissipation as it will affect the results. But we will be doing trials for each concentration and building a calibration curve. So and to have more insights on how the sensor is working when combining a microRNA on its surface and when there is a binding event on its surface.
And can you compare to the state of the art in terms of research?
Yes.
What has been done so far?
So far, for detection of sepsis, there is no QCM sensor related to detection of sepsis. However, in the literature, they can manipulate using microRNA on the surface of the sensor, QCM sensor especially, to detect other diseases. But could be that microRNA that got upregulated or downregulated in sepsis can be also correlated to other diseases. So we can tell, so no, yes, it is.
Okay, awesome. Thank you.
You're welcome.
Any other final question?
What about cost? The two methods.
Yes, so our technology will be cost effective for sure, because admitting patients to hospitals and while awaiting for the blood test to be achieved, the patient should be in the hospital. So there is so much money to be given in this stage, rather than using a sensor that will detect a sepsis in a matter of 1 hour or 30 minutes or 1 hour. So the admission of the patients in the hospital will be reduced, and then the cost will be also reduced.
Okay, thanks, Khulud. So our next speaker is Dana Bekdaj, who is a master's student supervised by Professor Jason Amatouri, and her talk is about tango of the hyoid bone and trachea against upper airway collapse in obstructive sleep apnea.
So I'm going to get straight into this. What is obstructive sleep apnea? Simply put, it's a disorder whereby the upper airway becomes obstructed repetitively during sleep. And as we can see in this image over here, we have an image of a person that's laying down. It's a sagittal cut view, and you can see here that the airway has been obstructed, and so this person cannot breathe. The air is not entering his lungs. So this presents several serious health concerns, such as daytime sleepiness or even reduced quality of life. But it can even go as far as causing cardiovascular diseases or even neurocognitive disorders due to a lack of adequate oxygen to the brain. So what causes this? Why do we have sleep apnea? Several factors can lead to OSA, which is obstructive sleep apnea. One factor is the anatomical factors of the upper airway, such as the hyoid bone and the trachea, which I'm about to explain shortly. So the hyoid bone. This is a bone that's located at the front of the neck. It's a mobile and floating bone that's connected to your airway with muscles, ligaments, and cartilage. And so it's essential for breathing, jaw movements, swallowing, and more. Now, for OSA individuals, it's common, as we can see in this image over here, to have a hyoid bone, which is this in yellow, to be lower than in healthy patients, which you can see up here. And this results in the airway to be less stiff and more collapsible. And so surgical hyoid bone repositioning has been a viable solution in order to improve the upper airway. But it's not that effective, with rates only between 17 to 78%. But the hyoid bone position and direction is also what makes the efficiency of the surgery possible. So next we have the trachea. And this is basically a windpipe, so it's a tube in the upper airway that takes air into your lungs when you're breathing in. It plays an essential role in maintaining your upper airway structure, as well as maintaining the soft tissue stresses. So its position can be impacted during sleep, weight loss, even breathing. But also, research has found that when the trachea moves downwards, which is when you're breathing in, it actually causes the hyoid bone to move downwards as well. And this is because they're actually anatomically connected. So we know that the hyoid bone and trachea are connected. We know that both of them are important. And so this makes it imperative for us to understand the interplay between hyoid bone repositioning and tracheal displacement in order for us to improve OSA treatment and understand the mechanisms of the airway better. So studying this in a physiological model is challenging due to the complexities of the upper airway. So we've studied this using a computational model of the upper airway of a rabbit. So why a rabbit? For two reasons. The first of which is the anatomical similarities between the upper airway of a rabbit and a human. And the second is because our lab has access to rabbits so that we can validate between the experimental and computational model. So this makes our aim of our research to simulate hyoid repositioning and tracheal displacement alone and in combination in order to study upper airway collapsibility, size, and soft tissue mechanics. So here we have an image of the model. It's a 2D model with 3D functionality. It is the upper airway of a rabbit in the mid-sagittal view, so cut like this. This model is made up of soft, bony, and cartilaginous tissues. We can also see different components of the airway, such as the tongue in red, the jaw in gray. We have the airway, which I've highlighted in green. This is the hyoid bone, which is this gray circle. Sorry. So this is the same model, but here I've indicated the muscles which are represented as springs. And over here we have this base plate, which is basically going to be displaced downwards in order to model the effects of tracheal displacement. And so we start off with a tracheal displacement downwards from 0 to 3 millimeters, then a hyoid repositioning from 0 to 3 millimeters in 5 directions, anterior forwards, cranial upwards, caudal downwards, and anterior cranial and anterior caudal in between. And finally, we have the upper airway pressure, which is a negative pressure within the airway, which basically mimics the negative pressure you experience as you're breathing in. And this will tell us more about the pressure that leads to the collapse of the upper airway, which is P-close, and gets us the area and soft tissue of the upper airway. So here we have an example of the simulation of the model. We've applied hyoid repositioning upwards, so in the cranial direction, tracheal displacement, and then upper airway pressure. And at the point where the airway collapses, we can get that pressure to get P-close. So here we have the percent change in P-close as a function of the tracheal displacement, and the P-close is relative to the pressure when we have no hyoid repositioning or tracheal displacement, so natural. As we're increasing our tracheal displacement, our change in pressure is reduced. What does this mean? It means more negative pressure is required to collapse the upper airway, so your airway is less collapsible. Then as we're increasing our hyoid repositioning, so in the anterior direction, for example, so from 1, 2, and 3 millimeters, we realize that the P-close is further reduced, so the airway is even less collapsible. And the combination of both tracheal displacement and hyoid repositioning gives us the best outcome. As for the cranial hyoid repositioning direction, same pattern, but the difference between the combinations is less significant compared to anterior. Now, meanwhile, for caudal hyoid repositioning, it actually has an inverse effect. So here, the tracheal displacement is reducing the P-close. However, when we're including hyoid repositioning, it's actually making the airway worse. The airway is becoming more collapsible. And you can see the maximum hyoid repositioning line in orange is now at the very top. Next, for anterior cranial and anterior caudal directions, it's the same pattern we noticed in the first graph, where the anterior cranial hyoid repositioning had the best effects on the upper airway, where the upper airway was less collapsible. As for the size in the upper airway, we notice here that as the tracheal displacement increases, the area, so the size is also increasing. And when we combine both hyoid repositioning and tracheal displacement, we're able to improve the outcomes even further. Cranial hyoid repositioning, again, same as anterior, but less significant. As for caudal hyoid repositioning, similar to the P-close value, it was an inverse effect. So again, your area is increasing with tracheal displacement, but as you're adding hyoid repositioning, it's actually making your area worse. It's decreasing your area. So here, the orange line is at the bottom. As for anterior cranial, anterior caudal, same pattern as the anterior direction, which is the first one, where the anterior cranial hyoid repositioning improved the airway even further than all the other hyoid repositioning directions. So here we had the greatest increase in area. Now for studying the stresses on the soft tissue of the upper airway. So stress is the measure of the internal loads that's located in the upper airway within the soft tissues of the upper airway. So the soft tissues are in blue. You have your stress from minimum to maximum, blue to red. When we include tracheal displacement, the distribution of stress is along the airways of the walls of the airway. Adding hyoid repositioning also increases the distribution of tracheal displacement, but now it's around the soft tissue of the hyoid bone. And then the combination of both further increases the stress in the soft tissue, and this increase in stress also results in an increase in stiffness of the upper airway. And this is the same pattern for all, where the combination of both improves the stress even further. And the direction of hyoid repositioning is what affects the stress the most. This is what makes the increase in stress most effective. where we had the anterior cranial direction, yet again, having the most efficient outcomes on the increase in stress. The strain, on the other hand, had the same pattern that we noted for stress. So in summary, tracheal displacement and high repositioning in combination and separately reduced peak lows and increased our size and stress and strain. And the combination of both is what helped make this even better. And so what does this mean? This means our airway is less collapsible and more stable, and especially in the anterior cranial high repositioning direction. So this research is the first of its kind to assess both tracheal displacement and high repositioning effects on each other and on the airway. And the significance of the research lies in its potential in order to translate it clinically with two strategies we've identified. The first is optimizing surgical hide bone repositioning based on a patient's natural tracheal displacement. And the second is by pairing surgical hide bone repositioning with stimulating tracheal displacement by muscle contraction that can pull down on the trachea. And so to conclude, this research provides proof of concept evidence of the fact that the tracheal displacement can further help improve the hide bone repositioning surgical outcomes through a combined approach. Thank you.
But you said you have used a rabbit but you have done only modeling, right? So why not humans in this case?
So because this is very invasive, it's very difficult to convince someone to just come, like open him up to try to, work with the airway. So because it's invasive, we want to try to reduce the amount of invasive work that we're doing in animals first, because the first stage is always through animal testing before going to clinical testings with humans. So we want to try to reduce this invasive nature by using computational modeling first, and also because by using computational modeling, we're able to control more factors that we can't do in a physiological model. So this way, we have this model which has already been validated through previous studies. We already know that it works and has been validated with a physiological model of a rabbit. So clinical testing on rabbits has been done to verify the model. But to simulate both tracheal displacement and high repositioning together hasn't been done on a rabbit. It's been done on this validated model instead.
Do you think that there's other effects on tracheal advancement or high repositioning have on the rabbit other than expanding of the tracheal airway?
So of course, since the physiology and anatomy is it's very complex, everything affects everything. Everything can have a different outcome depending on each case, let's say. But from previous studies, there has been a consistent findings of this increase in the airway due to tracheal displacement, this increase in the airway due to the position of the hyoid. It's always been consistent. And so this research has to study the effect of each on each other, given that, as we said, there's an anatomical connection. The position of the trachea does affect the hyoid. And so by studying these two, at least we get to understand this more controlled, let's say, experiment of how these affect each other and the airway, since we know that there is this increase in airway size, etc.
Yeah, but does it have other effects on the overall mouth of the rabbit?
By tracheal displacement, you mean?
Yes.
So the primary, other than the fact that the trachea obviously helps with breathing, but the primary factor that happens during breathing in, as in tracheal displacement downwards, is it stiffens up the muscles, which does pull the airway open. If, for example, there is a case where the airway doesn't open enough, it could be related to external factors, not necessarily the trachea. Then it becomes a more complicated system. But we do have proof, and we do know that from the way the upper airway works, by pulling the trachea downwards, the muscles are becoming stiffer and is supposed to support the airway to open.
Okay, thank you.
Thank you, Hiba. I think we have to move to the next presentation. Thanks a lot. So our next speaker is PhD candidate Zena Halabi, supervised by Professor Masoud Khreish, and her presentation is on single-cell biomechanical profiling of the metastatic potential of MDA MB231 cells. So I'm curious to know what that's about. So you have the floor. Thank you.
Good afternoon. My name is Zena Habli. I'm a PhD student in biomedical engineering, and today I'm going to talk about mapping the metastatic potential of cancer cells through their mechanical properties. So breast cancer is the most common cancer in women worldwide, and it accounts for more than 40% of cancer cases in women in Lebanon. We, women, are advised after a certain age to do some self-examination on a regular basis to check if there are some possible lumps in our breast tissue. And if we feel something that is worrisome, we would be advised to go to the physician, get checked by palpations, and then go and get a mammograph. This palpation examination gives an impression that the tumorous tissue or the possible lump is cancerous, is stiffer than the normal tissue around it. But the question really is, are cancer cells really stiffer than normal cells? In order to understand this, we gotta go back to the basics and understand how cancer happens. Upon cancer initiation, a cell undergoes a complex functional transition called EMT, or epithelial to mesenchymal transmission, where the cells will lose their normal epithelial phenotype and start acquiring mesenchymal cancerous phenotype. This happens under the influence of gene regulations, where we have upregulation, downregulation, and even inhibition of certain genes, such as fibronectins, cadherins, cytokeratins, connexins, among others. And this genetic alteration is also accompanied by by cells deviating from their normal mechanical homeostasis, where remodeling of their extracellular matrix starts to happen. The cells start losing their cell-to-cell contacts and cell-to-underlying substrate contacts, and they become able to be motile and migrative. EMT is believed to be the common cause for cancer spread in the body in a process called metastasis. And for metastasis to happen, from the primary tumor, some cells should shed, circulate in the blood, and spread the disease into the body. For a cell to shed, it has to be very elastic in order to detach from its primary tumor. It has to be very viscous in order to withstand the shear forces of the bloodstream, and very soft in order to evade the immunity. And if all of these combination happens, the circulating tumor cell will, or the CTC will survive the bloodstream, and it will spread, and it will create distant or metastatic tumors in secondary sites. So therefore, identifying, characterizing, and quantifying these mechanical signatures of CTCs, basically their adhesive strength, elastic deformation, and viscoelasticity, will give us an insight about the degree of cell invasiveness of these CTCs and subsequently their metastatic potential. There are a lot of studies that have investigated the mechanical properties of cancer cells, but most of the time this has happened by using different cell lines that have different genetic background, and thus the results that are reported are usually influenced by the genetic lineage of the cell itself. In order to resolve this issue, by the help of Dr. Saban from the Faculty of Medicine, we have developed our own cell model that has a different or a spectrum of metastasis by upregulating and downregulating a gene called connexin 43. When upregulating connexin 43, the cell becomes less metastatic, almost non-tumorigenic or normal, and we will be referring to it as connexin 43 dendra cells throughout the presentation. When downregulating connexin 43, the cells become highly metastatic, highly motile, and we will be referring to these cells as SH connexin 43. In order to study the adhesion potential of the cells, we applied a protocol called single cell force spectroscopy, where it's usually, it depends on atomic force microscopy, where a cantilever is coated with something in order to handle the cell, and then the cell will be allowed to interact with underlying cells or materials in order to study their adhesion potential to the underlying substrate. However, AFM is very cumbersome. It gives low resolution data. So we moved to, we leveraged the technology called fluid FM that has, that combines atomic force microscopy with fluidics. We have a cantilever that is hollow and it is filled with liquid. This cantilever is allowed to approach the cell, immobilize it with negative pressure, and then pulling it out in order to study its force of adhesion. Fluid FM technology gives high resolution. The forces of adhesion that we measure are in the orders of nanonewton, where typically AFM gives us data in the order of piconewton, and it is high throughput. Here are some images, real-time images of a cell being pulled out by a fluid FM cantilever. And this happens in a matter of 1 minute. The forces, if the cell that we choose is a single cell, then the force of adhesion we are measuring corresponds to the force of adhesion of the cell to the underlying substrate. But if the cell is surrounded with adjacent cells, then the force of adhesion that is measured has an added component of cell-to-cell interactions. And upon the movement and the retraction and the deflection of the cantilever, a laser would be recording these movements, and we will be digitizing this signal into what we call the force distance curve, and we can calculate the force of adhesion based on Hooke's law. Now we move to some of the results. We compared single cell versus cell in a cluster force of adhesion, and you can see that there is a major evolution of the FD curves from the single cell state to a cell in a cluster state. Most interestingly, the connexin 43 dendra cells that are the non-metastatic cells, they possess the largest forces of adhesion, followed by the wild type, and then followed by the highly metastatic SH connexin 43 cells. Another interesting result that we found is that these differences between the different cell lines, they vanish when we study the cells in their single cell state. Then we wanted to study the elasticity of the cells. So we moved to studying the spring coefficient of the cells by dividing the force of adhesion to the distance traveled by the cantilever in order to fully detach the cell. And similar to the adhesion pattern that we have seen, the highly metastatic cells, they were the highly elastic. Then we performed some QCMD studies in order to study the viscoelastic character of the cell membrane of the cells. As my colleague previously mentioned, we have a sensor that oscillates, and when we add or remove masses, we will be seeing a shift in frequency, but this happens only in dry conditions. When working with cells, we are introducing the shear modulus into the equation, and thus we will be able to analyze the viscoelastic character of the cells as well. And since we're already measuring the dissipation, we can get more information about that. For the sake of time, I'll move directly to the results. As you can see, upon examining the frequency and the dissipation, we have a single-phase QCMD response for the frequency and the dissipation in all the three cell lines with a sharp increase in the frequency and a sharp decrease in dissipation, but with different levels when comparing the different metastatic states. The highly metastatic cells, they showed almost double the results in both the frequency and the dissipation, showing that they are more viscoelastic, and this was also confirmed when analyzing the dissipation frequency curves or the DF plots. The highly metastatic cells had the steepest slope, and thus this confirms that they are viscoelastic. So with this very brief results that I have presented and many other experiments that we have performed, we can confidently say that the cell mechanics can be mapped with the metastatic state of the cell. The highly metastatic cells, they are less adherent, more motile, have a viscous membrane, and they are more elastic. This is very important because high levels of cell invasiveness always generates permissive conditions for CTCs to form, and these CTCs will be highly invasive and thus spreading the disease into the body. And understanding these mechanical evolutions of the cancer cells will help us estimate the level of the cancer invasiveness and even design tools or sensors that are capable of detecting circulating tumor cells from a blood sample based on their very unique mechanical properties for diagnostic purposes. And I'd like to acknowledge or thank my supervisors and the EUB funding bodies for supporting this research. And I think that's it. Thank you.
A lot. Questions?
Hi, thank you, Zena, for a lovely talk. I'm not too familiar with Fluid FM, but just by looking at it, won't it matter where you put the cantilever on the cell? I mean, the location of the cantilever, won't that determine how easy it is to rip the cell off from the surface? Is that controlled in any way?
Yes, it is. Okay, so with the fluid FM technology, we have tips that have different sizes, 2 micrometer, 4 micrometer, and 8 micrometer. In my case, I was always working with the 8 micrometer, and I was positioning accurately the interface to target site between the cantilever and my cell at the nucleus in order to fully detach the cell and get an idea about the total adhesion of the cell to the underlying substrate. If we want to study other adhesion components like the focal adhesion points and other stuff, we would be using smaller tips or smaller apertures, and we will be targeting them at the let's say, the boundaries of the cell or the rim in order to study this specific interaction. I hope this answers the question.
All right, thank you.
Other questions from the audience? Pardon.
Elena, was a great presentation. Can you comment on other common techniques that are used to measure mechanics in single cells, and whether they are, superior or similar? What are the advantages of the fluid FM compared to others?
Okay, so the first one is AFM again. It has low resolution and it takes a lot of time, and we're always coating the tip, and thus we are introducing more factors that will affect the results that we got. I know for a fact that they use the traction microscope. They also use it's called DF. It's like a flow cytometry where they will be pushing out the cells in single cell states, and they will be measuring the resistance to the flow. This needs a lot of time to let's say, iterate and optimize the flow that is capable not to kill the cell and not to flush it away while recording their elasticity. All of this, again, is fluid FM is expensive. I cannot say they are less costly. But again, it's all about the time and being able to recover the cells after working and keeping these cells alive if you want.
Thank you so much, Zena. The next speaker is Rayyan Sayed Ali. Is she here? Okay. She's a PhD student working with Professors Joseph Constantin and Yusuf To. And the title of her presentation is Instantaneous Creatinine Testing in Urine Using a Modified Bipolar Helix Antenna. Okay.
Hello, everyone. So this is Rayyan Sayed Ali. I'm a PhD candidate at the Impact Lab, working with the supervision of Professor Joseph Constantin and Professor Yusuf To. Today, I'll be presenting about our research work, which is about instantaneous creatinine testing in urine using a modified bipolar helix antenna. I would like to mention that this work is now under provisional patent protection, in addition to that this work is being conducted in collaboration with the Faculty of Medicine here at AUB and the Faculty of Medicine at USG. So before we dive into the details, let's take a moment to outline the progression of this talk. First, I will be delving into a background, and then I will break down the topic into several main points, including the sensor design, the preclinical testing, the sensor performance evaluation, in addition to animal testing, clinical testing, and I will wrap everything up with a concise conclusion. So as we know that monitoring biomarkers in blood, in urine, in tissues, or any other body fluids is essential to be able to assess the health condition of any individual. Usually, patients do this assessment of their circulating biomarkers either at medical laboratories or at hospitals. However, monitoring or measuring biomarkers at laboratories and hospitals requires specific sample collection logistics in addition to specialized equipment and reagents. It also requires technically trained workforce in addition to being timely consuming. Now, chronic kidney diseases are a growing concern affecting nearly 35.5 million adults in the United States. They are also associated with increasing morbidity and mortality rates, and they are posing a global and significant healthcare challenges to the worldwide healthcare programs. Usually, the creatinine biomarker is measured in order to be able to assess the kidney functioning for us to be able to pause or slow down progression into kidney failure. And the testing methods that are often used to measure this critical biomarker are either a blood test to measure the glomerular filtration rate or a urine test to measure the albumin to creatinine ratio. However, all of these testing methods are chemical. They are pH and temperature sensitive, in addition to being costly and time-consuming. So in this work, we are proposing a novel sensor design that is capable of addressing these limitations effectively. This sensor is instantaneous, chemical-free. It is able to provide an accurate estimation of the creatinine constituent in a urine sample. In addition to that, this sensor transports the facility of testing, the medical testing facility, into the comfort of the patient's home. In addition to this, it is capable of offering an approach to medical and point-of-care applications. So here I'll be discussing the main features of this design. The design is basically designed as per the bifilar helix antenna structure. It is a bifilar helix with 2 arms. Each arm is constituent of 3 turns. And then we have a PLA plastic column that separates this bifilar helix from a second slotted metallic cylinder. And these slots are inspired by the complementary split ring resonator design. These slots help us accentuate the sensitivity of the bifilar helix antenna to any changes in the creatinine constituent of any urine sample that is loading this sensor. At the core of the sensor, we have a plastic column that holds the urine sample under test. In the figure to the right, we show the electric field distribution inside the sensing area, inside that tube that holds the sample. The highly concentrated areas, which are shown in red, are designated as highly concentrated areas with the electric field and thus highly sensitive to any changes in the constituents of the loading sample. So after fabricating a prototype of this design, we did some standalone measurements in order to be able to compare simulated data with the measurement results, and a great matching was observed here. The sensor, as we can see, operates over the frequency band ranging from 4 to 6.5 gigahertz, and it is characterized by its volumetric sensitivity that is capable of sensing any changes in the creatinine constituent of the loading sample with high sensitivity. The sensor is also a multi-band sensor, which is characterized by a multi-band response due to the slots that are designed inside the metallic cylinder, which is the 3rd layer of our sensor. So basically, this sensor, it leverages the interaction between the electromagnetic waves that it emits and the loading sample of urine, and then it extracts all the physical S parameters of the system, extracts all the system extracts all the physical S parameters of the sensor that are being changed upon the change in the concentration of creatinine inside the urine sample. After that, all of this data is being pre-processed, and regression modeling is applied to this data in order to arrive at a model that is able to translate the S parameters of the sensor into a readable response of the creatinine level in the urine. So now that we discussed the main features of the sensor, we now move into a critical step of sensor performance validation, which is the preclinical testing. So in this experiment, we used artificial urine samples that were prepared in the normal and abnormal range of creatinine, and these samples were loaded one after the other into the sensor, and using a vector network analyzer, we collected the S parameters of the sensor upon loading these samples. This figure to the right shows the reflection coefficient of the sensor and how it changes for each of these samples with distinct creatinine concentration. So after the collection of the S parameters, this data is being pre-processed, and the pre-processing step is usually done through averaging, sampling, and normalizing. And the output of this stage is being fed into a wrapper, a forward feature wrapper selection method, which is capable of identifying the critical features or frequencies that are highly sensitive toward changes in the concentrations. Now, the output of this stage are fed into a regression model in order to be able to estimate the creatinine concentration in each sample. Here, this figure shows the high correlation between the S parameters and the concentration, and the other figure shows the high accuracy in predicting the concentration of creatinine. In this section, we did some performance evaluation upon spiking the samples with some protein interference that are naturally present in urine. And by using classification techniques, we were able to classify normal from abnormal samples with high accuracy of 93%, sensitivity of 91%, and specificity of 97.5%. And now we use 12 control rats in animal testing and 12 type 2 diabetic rats in order to be able to show the accuracy of the sensor in determining creatinine in their urine samples. We collected urine at baseline after 1 month and after 2 months of diabetic onset, and in the 3 time frames, the accuracy was above 90%. Now, concerning clinical trials, we used the urine samples collected from 42 patients, 23 of them are control, and 19 are with type 2 diabetes. The same experimental and data analytics procedure is repeated, and this figure here shows that the predicted concentration using the model that we built, the regression model that we built, is in high correlation with the reference creatinine concentration with an accuracy of 93%. So I would like to re-mention what we did in brief. We designed a novel RF sensor that is capable of accurately detecting slight changes in the creatinine concentration of a urine sample, and we conducted preclinical, animal, and clinical trials and validated that the accuracy in the 3 of them is higher than 93%. Thank you.
Thank you very much.
Thank you for the presentation. Just a simple question regarding the frequency range. Why did you design your sensor for this frequency range exactly?
So basically, we measured the dielectric properties of artificial urine samples spiked with different creatinine concentrations. And by measuring these dielectric properties over a huge range of frequencies, we were capable of identifying the most sensitive range towards these changes. So this microwave range from 4 to 6.5 is characterized by a high sensitivity towards changes in these concentrations. That's why we were sure now that the sensor will be highly sensitive if it is designed in this range of operation.
Can you conceptually weigh in some remarks on why would you need a specific antenna designed to a specific sensing application? Like suppose you have an antenna that can transmit in a broadband, entire frequency band. What are the links between the specific sensing application and the constraints on the design of the specific antenna?
Okay, so basically, the most important feature of the sensor should be its sensitivity to the changes in the specific biomarker that we are targeting. Now, after identifying the suitable range of operation, we will be designing the circuit that we need, either an antenna, either a resonator, either a filter antenna. So that is basically determined based on, first of the sample that we can use, how big the sensor can be, the volume of the sample. In addition to this, the number of resonances that we need in order to have critical features. So, for example, in this design, we were able to achieve a triple band response, which ensures that we have changes at different frequency, and thus we achieve higher sensitivity.
Just one question. If it happens that another sample resonates, is that the correct term, resonates at the same frequency that creatine resonates at, what would you, how would you avoid the confusion between which biomarker is causing the shift?
Yes, so basically we conducted an experiment that is exactly targeted for this conclusion. So we spiked the sample with different proteins that have the same nature, which means technically the same dielectric properties of the biomarker under test. And we were able to show that the specificity of the sensor to detecting creatinine is 97.5%.
Okay, so creatine. So the frequency you're looking at is only true for creatine. It's like a fingerprint for creatine. There isn't another biomarker that has the same.
It might be for a single frequency. However, here we're working on a range of frequency that measures over 202 frequencies. And the model is not over one frequency. So the model works on several features. Sometimes it's 15 features. It might overlap for one frequency, two frequencies, but at the end, when you are modeling using the whole critical features that you identified, it won't be exactly the same.
Thank you very much. So we'll move
you.
on to the next presentation.
Okay, so our next talk for this session is gonna be given by Ghadir Jalloul. She's a chemical engineering student, PhD student, under the supervision of Professor Muhammad Ahmed. Her talk is on TiO2, PVDF, HFP, electrospun membranes for tetracycline, antibiotic photocatalytic degradation under visible light. So I'm quite interested to know what all this means.
So hi everyone. Today I'm going to talk about TiO2 immobilized over PVDF HFP electrospun membrane used for the removal of tetracycline antibiotic using photocatalytic degradation as a technology. In the beginning, I'm going to explain what antibiotic resistance is, then move to talk about the different removal technologies and how we used electrospinning technology to prepare our membranes, and how we tested those membranes, and how we reached the conclusion that we need a totally new design. Starting with antibiotic resistance, it is actually the presence of superbugs or resistant microorganisms that have developed resistance toward specific antibiotics in a way that they are no longer affected by these antibiotics when they are treated. So they continue to grow and develop and cause more illness. The reason behind that is that they are continuously exposed to antibiotics in the environment as a result of the overuse and misuse of antibiotics that are eventually discharged in the environment. As you can see, based on these statistical data between 2000 and 2030, there is a huge increase in the consumption of antibiotics. The reason why the WHO has classified antibiotic resistance as a global crisis is that it causes incurable bacterial infection, increase in patient mortality, escalating treatment costs, and outbreaks of diseases. There are different sources of antibiotic contamination or pollution into the environment, from houses and hospitals, from agricultural and livestock industry, as well as pharmaceutical production. Because the current wastewater treatment plants are incapable of fully removing antibiotic from the water streams, there have been several new technologies, as listed here, that can be used to totally remove antibiotics from the environment. We focused on advanced oxidation processes, as they are more eco-friendly and produce no secondary pollution. And out of them, we focused on photocatalytic degradation as it can be operated under solar irradiation. It can be operated under atmospheric conditions. The antibiotic we used or studied was tetracycline, because it's the second most used globally, as it's being cheap and has wide antimicrobial spectrum effect. As photocatalytic degradation says, it needs photo energy, which is light, and it needs a catalyst, as will be explained in the next slides. The methodology we followed was to prepare our membranes, characterize them, and test them. The question would be why we need membranes. So we need a catalyst because it's photocatalytic degradation. And then because we aim for a large-scale application of photocatalytic degradation. We previously used nano-sized and micro-sized photocatalysts, got super good results, published works, and so on. But photocatalytic degradation by itself has not been industrialized up till now. So in the studies to industrialize such a technology, one thing was to facilitate the recovery of the photocatalysts from the reactor by immobilizing those nano-sized or micro-sized catalysts on fibrous membranes. That will be shown in the next slides. So the technology used to prepare our membranes was electrospinning. As you can see, it has high voltage power and zero voltage on the other side. So the difference in high voltage is the driving force for our fibers to be produced. Rather than only electrospinning polymeric solutions, we added our TiO2, which is the photocatalyst, into the polymeric solution to eventually get fibers that are decorated with the photocatalyst and just put our membrane in the solution, the reaction happens, and we remove them by minimizing the cost and time to recover catalyst from pure water. But back to photocatalytic degradation. As I said, it's a technology that requires light and a catalyst. A catalyst is added to the system. It can briefly be described as antibiotics in the system. In the presence of light and TiO2, which is our photocatalyst, antibiotic will be converted into H2O and CO2. So from a toxic pollutant to just CO2 and H2O in a mechanism that can be classified as excitation of electrons, production of radicals, and then degradation. Back to the catalyst. So this is the nano-sized and micro-sized catalyst, TiO2. It was electrospun. Those are the pivot HFP polymeric membranes. So using electrospinning, we got fibers decorated with TiO2. So now my catalyst is on the fibers of the membrane. In our research part, we developed the best method to prepare the membrane or combine the photocatalyst with the polymeric solution in such a way to minimize any precipitation of the catalyst in the polymeric solution by dividing the solvent into two parts, dissolving the polymer first and then dispersing the catalyst in the other part and combining them. We also varied the voltage for the by changing the parameter of the electrospinning and got the best voltage as 14 kilovolt. As mentioned before, we mentioned there are various parameters for electrospinning to be controlled. After that, for the testing of our membrane, this is our conventional system, or the system used in various studies to test nano-sized catalysts, where it has a reactor or a beaker, a magnetic stirrer, and the light above the beaker. Traditionally, we just added our catalyst, the polluted water, and we turned the light on and record the degradation of the antibiotic. However, now we just added our membranes into this system because we immobilized our catalyst on those membranes. Unfortunately, when we were observing, the membrane was floating on the surface of the water or just being folding. It was folding, which means it was not fully exposed to the light that is put above the system. Because we were limited in the system, this is what we used previously in all our studies, we tried using 3D printed membrane holders to force our membranes into the system by using different surface area and different geometries. At the end, we got really low efficiency as compared to what we got previously with the nano-sized and micro-sized photocatalyst, the work we published, and so on. But because we really want to move to a next step of industrializing photocatalytic degradation, we want to change the whole system. So using the membrane with the conventional system results in low efficiency. There is a need to develop a new system. The new system was this design. It's a closed system, and rather than putting only one part of the membrane and being limited with the amount of catalyst found in the system, we designed this membrane holder where you can cut your membrane and attach different and larger area of the membrane all over this membrane holder. And by this way, we minimize the distance between the light and the membrane, In this way, the membrane is being irradiated from two sides rather than only from the top. And the membrane is being rotated externally rather than the solution being rotated. So in this way, we increase the visible light or light absorption by the catalyst on the membrane. We minimize the distance. We minimize the requirement of the spatial requirement to put more membrane and more catalyst into the system to get higher efficiency. That's it again. Using the conventional system of photocatalytic degradation with the membrane is not efficient. We really need a new design to account for the low visible light absorbance and to maximize the area of the membrane and minimize the spatial requirement. And that's it.
Hello. So how much time does it need for the antibiotics to fully, and also what's the benchmark of how much antibiotics are left in the solution for it to be safe?
I'm gonna compare our results for the published work we did already using the nano-sized TiO2, all right, which is the photocatalyst, rather than immobilizing them. In the first published work, we reached 100% removal in just 80 minutes. Okay,
80 minutes?
So 80 minutes, it was fully removed.
How much solution did you put inside?
The concentration, it was 200 milligrams of tetracycline in the solution. The volume was around 300 milliliters.
So if you increase the volume, it would require more time, right?
It depends. It depends. So
Depends on the concentration.
Depends on the whole system. So maybe you will need more power of the light. So the light is up. You are slightly limited. So the light is up. You will need maybe more catalyst, maybe higher intensity of light. We studied actually the intensity of light, the effect. As you increase it, of course, more energy will be put, so more excitation.
Thank you.
You're welcome.
More questions?
So how does this compare to other systems that are published already?
Well, in photocatalytic degradation, there are various things to be optimized, starting with the reactor design, the catalyst by itself. So our work, for example, was based on modifying and increasing the visible light absorbance of the catalyst by itself. So rather than working on the system itself. So now we reached an optimal catalyst based on our work, previous work. Now we took another step to help industrialize it because based on his question, okay, I want to put like 1,000 liters in a reactor. I know my catalyst is super efficient, but I will need more and more weight of the catalyst to be dispersed. Okay, at the end, I got 100% removal. So I have pure water, but in those water, there is the catalyst. Now I have to separate the catalyst from the water. So it's not practical. There are many things to be optimized. So now I can get the optimized catalyst and then think about a way to help recover it from the system, which is this way. So when we reached this point, we figured out that, okay, the whole system should be changed due to the limitation of visible light and irradiation into the system and so on.
You said that we will be using 5 times more or like 6 times more antibiotics in 2030. How did you come up with that number?
This is based on this report. This report mentioned that.
How did they come up with the number that they are predicting that we are going to be using 6 times more?
It's a forecast. It's a forecast based on some modeling. In case no policies and restrictions were taken to limit the consumption in the previous years. So they projected it in a specific based on like medical issues and so on. Which is 10 times actually in 30 years, which is huge.
Very much. I think we have to move on to the following. Okay, so the next talk is by Andrea Tarabai. Actually, she's the only undergraduate presenter for today. She is an ECE student supervised by Professor Nassim Daher. She will be talking on object detection through a learning joint initiative supervised anatomy framework, GISA for short. So the floor is yours.
Hello, everyone. So my name is Andrea Tarabai, and today we're going to talk about object detection through a learning joint initiative supervised autonomy framework. So first, I'm going to ask you a question. What do you see? A pencil, right? Does anyone see it something different? No, it is a pencil. But do you know that actually goes through a process? It's not that easy. Our vision isn't that simple. So first, our eye captures the light from the pencil, and then it gets transformed into electrical signals, which then our brain processes it, and then the brain processes the features in the pencil, and then it makes a conclusion of, oh, okay, well, that's a pencil. So visual perception isn't that simple. Actually, it's really hard for like how it happened. The process is complex for humans. So how about if we want to implement visual perception to machines, right? So machines, to have visual perception, they need it because in order to act or plan anything in their environment, they need to see where they're going or the object, for example, they're about to grab. So as humans, we use visual perception to move in our environment and be able to interact. So in this cycle, first a machine or a human, for example, is in their environment. They use their perception to sense their environment, to see where they are. If they're going to grab something, for example, they see it, and then they plan how about to take the action. If they want to go there, what is my path? I want to grab it, how am I going to grab it? And then we do the control, we act. So we send signals to the joints, for example. We command our arm to go and grab this thing. And then the action takes place. So this cycle is a complete cycle. So what happens if one of them isn't that good? So object detection is one of the challenging tasks that computer vision requires to localize and classify objects. And as I was saying, what if the perception of a machine isn't that accurate? And why is that? Why would a perception of a model, for example, of a machine, not that accurate? Because it has limitations. And limitations include different object sizes, lighting conditions, positions, occlusions, meaning if objects are behind other objects, a model might not detect that object to be what it is. It might detect it as something else. And as much as object detection is advancing, it has not yet reached a point comparable to the perception of humans, limiting it to be free in its environment. So for example, here we see a cat, right? However, I tested it on a model, and it detected it as a person. And that is just because the cat was wearing a suit and tie, so it thought that the cat was a person. And surprisingly, in other models also, like this as a person. So sometimes different lighting conditions lead to wrong conclusions, which would break the cycle and would lead the robot to not successfully complete its task. Here, for example, also you see a spoon. Just because the background wasn't that clear, it had sharp edges, it compared that to be a spoon, but the other one to be, for example, a knife. So we know that there are many object detection models that have reached like very high accuracies. However, in certain instances, in like certain lighting conditions, these might still happen. We didn't reach 100%. So that's why in my research today, I'm going to propose, say, the importance of human in the loop. As much as machines have advanced, we still need a human in the loop to have the possibility to intervene in the process. So why is a human in the loop is necessary? Because a human, for example, when we're learning a certain task, sometimes we get lost, so we seek help, and a human comes and helps us, right? And also sometimes our capabilities are limited, so we don't know, and our supervisor notices that, and the supervisor comes and intervenes in our task. So there's two ways. Sometimes we seek help in our learning process, and sometimes someone helps us without us knowing that we are doing the wrong thing. And the same we're saying to a machine. And there are many human-in-the-loop approaches that include a human-in-the-loop in the process of a machine in its environment. However, I'm going to talk about JISA, which is Joint Initiative Supervised Autonomy. And what is the difference that it is other than other frameworks is that the autonomy, the levels of autonomy between the human and the agent are not divided equally. Okay, so JISA prioritizes the autonomous agent operation. So the agent operates autonomously, but asks for help, and it's complemented by human assistance based on either robot requests or a human judgment. So what does JISA has? JISA has two metrics. First, we see self-confidence-based assistance. Self-confidence-based assistance basically, as we said, when the agent is not confident enough about its action, so it queries the human for help. This is the self-confidence metric. The situational awareness-based metric is when the human supervisor notices that the machine or the agent isn't doing well in what it's doing, either due to limited capabilities or due to other problems or dangerous situations. And that's when the human can intervene in its action. The problem is with JISA is that they formally define the level of cooperation between the robot and the agent. However, they did not include a learning aspect in it. So they formally said, okay, we have this metric, a self-confidence metric, and a situational awareness metric. However, if the robot or the agent goes into the same situation again, when it fails to do a certain task, it calls the human to help and to help again. So it will not adapt to changes in its environment. The human will keep on supervising, which will not lead the object detection model or the machine to actually learn. So the objectives of my research is to elevate JISA into a learning framework and specifically an object detection application. So what we're going to do, I developed a graphical user interface that allows the human supervisor to monitor, correct, and intervene. And we add a learning JISA aspect to it. So an object detection model, most of them have the CNNs, which have an input layer, hidden layers, and output layer. The image as pixels gets processed into the network, which then produces an output, a prediction. So if you want to use a model from scratch, use a model from scratch, it will take a lot of time, and that's why we're going to use a fine-tuning approach, where we use a pre-trained model, and then we train our model on a specific data set, for example, and then we get a fine-tuned model. So how do we tune an object detection model from human feedback? So we were saying, to recap, we want to implement JISA, but a learning JISA. So how do I get this human feedback and let my agent to learn from this human feedback? And that is by reinforcement learning from human feedback. Reinforcement learning is basically based on reward. When a child does a mistake, for example, you punish that child or you show him how to do it. And that is what reinforcement learning is. So we're going to take that concept but implement it with human feedback and add to it JISA. So our state, our input image, where we are is the image, and the action at the end is the label and bounding box of what our image is. So when our model detects a label and bounding box, it gets a reward from the environment where we're going to use it as a human feedback. So the reward is from the human feedback. And what we use is iCreate. This iCreate is implemented using reinforcement learning from human feedback. It has interactive, corrective, and evaluative feedback. It has a self-confidence metric, which allows the agent to query the supervisor for help, and then the supervisor corrects the agent if needed. So I wanted to formally define JISA. Task to be supervised by the human is the object detection. The agent self-confidence attributes is when does the agent query the human for help. It is when the model is not confident about its prediction. So usually detections above 0.75 are deemed correct. So we're going to prompt human assistance when the confidence score is between 0.5 and 0.8. And the human intervenes when above the confidence score 0.8, the human intervenes to correct. So the JISA-based GUI that I did is one that has the model implemented in the graphical user interface, which, for example, the example we saw that detected a spoon and a knife. It wasn't confident, so it queried the human for help. The human can delete the box, for example, if it was a wrong box, or create a new box. And based on the human feedback, the model learns. And now we're in the process of getting the results, and we're training it on different data so that we see how a learning JISA improves the accuracy of an object detection model. So thank you.
We have questions from the audience?
Hi. Can I ask you about, you mentioned the example where if a child does something wrong, and you mentioned that there's the same example but for the model. So can you just go back to that slide and explain when you said that there's the reward from the human, what's this, what does that mean? What is a reward to like this interface? What's that?
So the reward is basically when the agent, for example, queries help from the human. If the human replies by yeah, it's a correct, you're not what you were not confident about, it's actually correct. It is the correct label. Then it is a reward from the human. So this is mapped into a reward from the human. But also, for example, in between, if the human comes and supervises the agent, the human can even validate the object detection model. So it can like give it +1. You're doing correct, for example.
And is there an opposite? So like if there's a reward, if they were unconfident about something and it turns out to be right, is there an opposite as well, like not punishment, but like if they do something wrong, is it just like so that the software doesn't come back and always keep asking the human? Like what's the equivalent, the opposite equivalent?
Yeah, the reward, and there is a reward and there's also a punishment. So when the agent queries the human for help, if it's unconfident, for example, the human either gives it a reward that you're unconfident but you did the right thing, or if it was unconfident about the label, then it would say no, you did the wrong thing and would update the label. So it's also evaluative from iCreate, as we mentioned, and also corrective. So it evaluated the query by saying no, you did the wrong thing, but also it then corrected the query by changing the label and the bounding box. So it either can change the label or even the bounding box. So it may have predicted the correct label, but the wrong bounding box. So it can also correct that.
Thank you.
Yeah.
Other questions?
Hi.
Hello.
Thank you. How is this different from supervised learning with a huge data set?
This is an online learning. It's continuous. So we're not just capturing the data and then training the model when the model is offline. Okay, here we're continuously teaching the model as it's going. That's the difference.
So that would be useful when, for instance, you don't have enough data to have a model.
Exactly.
So you would go you would learn as you go.
Yeah, it's in certain cases where it's a rare incident that happens. So you don't know when this happens. So that's why you keep the learning on. Like we're making the learning like a human learning. So the feedback is always there if it's asked for it. Yes.
Thank you.
Hi.
Hello.
Is it only done on pictures? Are you doing it on videos?
No, it's only on pictures for now, but of course it can be later on implemented on videos, because videos, it's just like collection of pictures. So yeah, of course it can be later on.
Hi, how
Hello.
are you? So I've noticed there's the object is within a box.
Yes.
Is there a way? So suppose there are two objects very close to each other and you cannot isolate them with the box. Is this causing any more confusion for the machine, or is there any way to actually, instead of using a box, like just tracing the outline of specifically that object?
In this interface, there isn't this feature, but usually the models take this specific model. It does not take a trace, because it's too complex for a neural network. It takes a coordinate of the box. Okay, so even if they are above each other, you can still put the boxes above each other. So you could place two boxes, as you see here, they're above each other. The mispredicted ones, but no, I mean, like, they can be implemented like that.
All right, thank you.
Thank you very much.
Thank you.
So our next talk is by Abbas Hassan. He's a PhD student in the AC department, supervised by Professor Sami Karaki. And the title of his talk is New Efficient Field-Oriented Control for Induction Machine Drives.
So thank you so much. I'm Abbas Hassan. I'm a PhD student. I'm working with Professor Karaki and Professor Bazzi. And today I'm going to talk about a new efficient field-oriented control for induction machine drives. Just first of all, I know that induction machine at AUB, they are not familiar with the students or any kind of machine in the graduate level. So I will start briefing with introducing the induction machine, what is the induction motors or the induction machine and how it works. So first of all, I want to mention that induction machines are widely used in the industry. So about 85% of induction machine motors used in the industry are based induction machines. Something machines like used in electric vehicles or in mixers, crushers, and compressors. So it's very important to look at these machines and try to just enhance the efficiency as much as possible. So that as a brief introduction of the concept of induction machines and how it works. So you know that three-phase system, you are maybe electrical engineer and you know that. So applying a three-phase system to a motor will produce some kind of field or magnetic field inside it. And this magnetic field is a rotating magnetic field, so it rotates inside the motor. Okay? This kind of rotor or resulted rotating rotor flux vector, which is rotating inside the motor, is the main variable that determine the efficiency of the system. So, my work in the PhD thesis. First of all, we have an induction motor, and this induction motor is put, let's say, in a vehicle, an electric vehicle. So, as you know, all you guys, you know the inverter from the solar inverter. So you control the induction motor by an inverter, a three-phase system, okay? In order to control the inverter, we use some kinds of controllers named FOC controller or the field-oriented control algorithm. The points here that how I have to control this FOC controller. So I have to put some optimal rotor flux value. This optimal rotor flux
is just this rotating rotor flux here. So I want to put a value that could be optimum for the efficiency of the whole system. So I tackled two main issues in this controller here. The first one is how to estimate. You know, this block is the the induction machine loss model. This block describes the kinds or the types of the losses inside the induction motor, which are the core losses, the copper losses, and the power electronics losses. How this box works. So first we need to estimate the speed and the torque of the motor using the currents and the voltages. We use this kind of nonlinear estimators just to estimate the speed and the torque, and we feed these parameters to the IM loss model. And this model or this box will determine the optimal rotor flux that can set for the FOC controller. So I tackled two issues. The first one, I invented a new loss model that wasn't founded before. And also I look at the nonlinear estimator types and I make a strong comparison between them. So this is actually the FOC controller. I'm not gonna go deep with the concept of FOC controller, but just I want to mention, so the two variables which are the mechanical speed and the rotor flux, they are feed in the controller, and the main rotor flux here, as I said, that it can determine the system efficiency, and an optimal rotor flux is a function of mechanical speed and torque. So for the optimal rotor flux, we have many models found in the literature. Okay, so I did a review of the models that are existed in the literature. These models are split into three types. These models, for example, are dedicated for, let's say, the power electronic losses, the iron losses, or the whole system losses. Many parameters or many criteria are introduced here. There are many, and I'm not gonna talk about all of them. I choose 4 models from this review here in order to show my work, how it differs from the work found in the literature. So, these are 4 models here utilized for optimization or for maximization the efficiency of the induction motors. Models that are based, as you can see, guys, on the torque and on the speed also. And there are some assumptions regarding the direct current and the quadrature current of the rotor and the stator of the induction machine. These models, they have many problems. They have issues, and we want to just tackle these issues. Here we can see these models plotted in the 3D plane. I want to mention 3 points regarding these models. First of all, these models don't take into account the core losses effects. It's very important just to induction motor to take the core loss effects in order to determine the optimal rotor flux. Also, the flux estimation error, I want to mention some point here. If you can see that all or most of the models are exceeding the 0.8 level. The 0.8 level is the maximum allowed rotor flux for an induction motor. So it seems that these models are erroneous and they are not working well. Based on an old paper that was discussing the iron losses behavior on induction machine in 1995. Okay, yes. So I got the modified assumptions here. These are the new assumptions, and based on these new assumptions, I defined the new losses, the new loss models that are different from others. So these are the new models that I invented based on the literature and on the issues that I put as an objective to tackle them. Just I want to mention that we have a limitation. This plane, the horizontal plane, is the maximum allowed flux that set for an induction machine. If you can see that the proposed model in the green color is not exceeding the plane, actually. However, all the others or other models, you can see that sometimes they are good. They are based on the operation conditions. So sometimes we can see that they are lower than the maximum allowable limit, and sometimes in different operation conditions, they are higher. So far, just we tackled this issue. We have another problem with the estimator, the nonlinear estimator. So I did some comparison between nonlinear Kalman filters and multiple model estimators, just in order to check the accuracy of the estimated value, the omega m mechanical speed and the torque. I want to mention some experimental results here. So just you can look at the currents, how they are noisy, and I got the estimates, the noise, and also I got the rotor flux value there, and I can estimate it. Also, I checked the uncertainty regarding the operation condition of the induction motor at low speed and high speed condition. Say that, so I did some experimental results regarding the whole idea that I'm working on. These are the estimates that I'm glad about, actually, and that's it. Yet no conclusions.
So maybe if you could, in few words, describe exactly the novelty in the work compared to other benchmarks, and maybe what would be a killer application of your proposal?
My novelty is all the used or utilized models in the literature, they are erroneous, they have some errors. The new model, it can operate regarding the torque and the speed for all operation conditions. That's a novelty. So I just invented a new model that it can work well for every operation condition in torque and speed. You can see the others, they are not well doing the estimation or the loss minimization.
For instance, the electrical vehicles. So how does this impact real life?
So let's say you have an induction machine, you have an electric vehicle, and you have lithium battery, which the capacity of this lithium battery, let's say 60 kilowatt hour, okay? The efficiency of the motor, let's say at some condition, let's say low speed condition and high torque condition, it's 60%. When I define this model and we apply it to the induction machine, the efficiency will be improved about 30%. It becomes about from 60 to 85, let's say. The increment in the efficiency will save the battery, and it will give you a longer distance just to travel. That's it.
Thank you very much. So last but not least, we'll be hearing from Zainab. She's also a PhD student in the EC department. She supervised Professor Yusuf To. The title of her presentation is Stretchable Multiband Radio Frequency Sensor for Strain Measurement.
Good evening, everyone. My name is Zainab. I am a PhD candidate in microwave and radio frequency engineering at the Impact Lab. And today I'll present to you our work titled Stretchable Multiband Radio Frequency Sensor for Strain Measurements. So the objective of this work is first to design a sensor that operates as a dual band pass and dual band stop filter. Then this sensor is optimized to operate under two different scenarios. The first one is a stretchable sensor for strain measurements. The second one, now if we make this sensor flexible, we can use it for muscle health contraction, for muscle health monitoring and muscle contraction monitoring. And under these two scenarios, the sensor is interrogated using a flexible backend circuit. So we want to design a strain sensor, but why do we want to design it? First, a strain sensor can be used for structural health monitoring and for pressure sensing in geotechnical engineering. Now, if this sensor was made flexible, so it can be worn by a user, it can be used for athlete fatigue prevention, for astronauts, as they go to space and come back, they lose a lot of their muscle strength. Same thing for seniors, they lose their muscle strength with aging. It can be used for rehabilitation after an injury and for monitoring cognitive disorders such as Parkinson's disease. So this is the structure of the filter sensor, and I will break it down for you. First, we have the inner defected ground structure, so it's inner DGS1 and inner DGS2. These DGS will capture the signal at a specific frequency when it passes from the input port to the output port of the transmission line. Here, that it will capture it at 2.27 gigahertz. Now, after the addition of the outer DGSs, the outer DGS1 and outer DGS2, the same phenomenon will happen. The signal will be trapped at 1.67 gigahertz. And we also have the U-slots capacitor. They are etched in the feeding line of the transmission line, and they are responsible for the band pass response of the sensor, as well as they ensure the stretchability of the whole structure. Now you can see that the sensor is made of three different layers. We have the upper conductive layer, which is copper, where the entire slots are etched, the DGS slots, the horizontal slots, and the U-shaped slots. The middle layer, which is Rogers material, and this layer is cut along the horizontal slots and U-shaped slots in order to separate it into three different parts. Finally, the bottom layer, which is silicon RTV, and we use it to place the different parts of the sensor and to make it stretchable. So these are the three parts I've talked about: the bottom W part, the upper W part, and the H part. And you can see that before stretching, the gap between the two W parts is named G slot, and the gap between the W parts and the H part is named as G cap. Now, after stretching the sensor, G slot will be increased by a value offset, and G cap will increase by half of this offset value. Now, we want to measure the sensor under different strain condition, under different stretching states. So what we did is that we added a mechanical fixture so we'll be able to test it. And this mechanical fixture, we've extended the W parts, drilled holes in them. We designed mechanical holders to secure the whole structure with nylon screws as well. And this is the fabricated prototype. The fabricated three parts, actually, the W parts and the H parts, as well as the mechanical holders. Then the whole structure was assembled together. This one is before stretching, and the right one is after stretching to a certain state. Now, as you can see from the tables, that the frequency is increasing for each state. And what do I mean by state? So for state 0, for example, we have an offset value of 0 millimeter, which is the initial state of the response of the sensor. The state 1 is when the sensor is stretched to an offset value of 0.5 millimeter. State 2 for an offset value of 1 millimeter. State 3 for an offset value of 1.5 millimeter. And as you can see, for different stretching state, we have an increasing frequency in the band pass and band stop operations of the filter. And here I'm showing a comparison between the simulated and measured as parameters at different states of the filter. This is just me. I took a picture of the sensor while I was trying to stretch it. See how it stretches. So, as I told you at the beginning of this presentation, the sensor, if it was modified to a flexible structure, it can be used for muscle contraction monitoring. And what we did here is that we replaced the Rogers substrate with a flexible PET substrate. We removed the mechanical fixture and we replaced them with an upper RTV layer for better mechanical integrity. We have also tapered the input and output ports of the filter to be able to connect it or integrate it with SCT048 SMA connectors. These are specific types of connectors. We can use them with flexible materials or flexible substrates. And this is the sensor modeled above a human arm. And you can see that the specific absorption rate of the sensor at the skin layer is equal to almost 1.46 watt per kilogram. And this is actually a value lower than the SAR value specified by FCC, which is 1.6 watt per kilogram. This is the fabricated flexible sensor before assembly, and this one is after assembly. So we've tested this sensor over 30 different volunteers, 15 males, 15 females. And what we did is that we placed the sensor on the volunteer's arm, and we've asked him to bend his arm into 4 different states: 0 degrees, 30 degrees, 60 degrees, and 90 degrees. And when this happens, when he bends his arm, actually the contraction of the muscle is changing. So here we can see for different angle, we have an increasing trend of frequency, which indicates a certain contraction. And to make this sensor, we took it a step further towards a standalone device, and we've designed a flexible backend circuit to interrogate the sensor and to measure the output power voltages of the reflected and transmitted signal. So from this table, you can see that the minimum power, here, the minimum power means a resonating frequency. So you can see that for the minimum power or the minimum voltage is changing for each state, which means we can detect the resonating frequency, thus a certain contraction state. And finally, we designed several prototypes. We've designed a stretchable sensor for strain measurements, a flexible sensor for muscle contraction monitoring. They come with their own backend circuit to have a standalone system that is tested over 30 volunteers. And I'm proud to say that this work is published in IEEE Journal of Microwaves, Wearable Flexible Radio Frequency Filtering System for Muscle Contraction Monitoring. Thank you everyone for listening, and I'm ready to take your questions.
Hi, Zainab. Thank you. Very nice talk. I just want to ask you a couple of things, I guess. So firstly, if I get this right, the RF part of this is that you're acquiring the data wirelessly, right? I mean, it's acting like a strain gauge, the sensor, isn't it? So the innovation here is that it's wireless?
It is like, it doesn't require any wiring because it comes with its own circuit that measures the strain intensity. So this wireless, it is first an RF sensor for muscle contraction monitoring with high accuracy. You see, it comes with 4 bands, which means better accuracy. See this? We have 4 bands for resonating frequency, thus better accuracy.
So can you just clarify that again? So what do you mean it's more accurate because of the 4 bands?
It's more accurate because we're detecting the change over 4 different frequencies instead of just 1.
And so also, I guess because you're on this plot already, so what is simulated versus measured? So where is the simulation being done?
The simulation was done, you mean the software?
So what is simulation? Yeah, so did you talk about the methodology for simulation? I might have missed it.
Right.
So what's simulated mean in this circumstance?
The simulation was done actually on a software named HFSS, and what we did is that we've designed this sensor, like I can show you here. And what we did on the software, we've changed the distance between the W parts and the W part and the H part. Here, the distance was changed like in a gradual manner, which is for value offset. The same thing for the gap between the W parts and.
Okay, great. Just one last quick one. You said this could be used in cognitive disorders or something? Yes. How? In what way?
Yeah, for example, Parkinson's disease, you know, they have like problems with their muscles and so on, so it can be formed by the patient, and the signal can be sent to the caretaker if anything wrong can happen, if anything wrong happens.
Can you give me an example? So what do you mean? So what exactly would this be useful for someone with Parkinson's disease? Or they're shaking more, or they're...
Their muscles were...
Okay.
You can use them to continuously monitor their muscles.
Thank you.
More questions?
Thank you for the presentation. So just one simple question. Why did you choose, how did you choose, the value of the gap?
Yeah, so actually in the simulation and design process, we simulated several gaps, and we found that the best sensitivity was for the gaps we taken, which is 0.35 ground plane, which is G slot, and 0.25 for G cap. So this is the most sensitive gap, which will give us the most shift in frequencies for different states.
Okay, thank you.
Thank you, Zaina, for this presentation. I would like to ask one question concerning the back-end circuitry that you use to measure the parameters of the sensor. How would this compare to a vector network analyzer?
Okay, first thing, this flexible back-end circuit is not a vector network analyzer. It measures power level or the voltage level for the transmitted and reflected signals. However, as it can detect the minimum power or the minimum voltage, we're good. It's sensitive. We can use it.
I see. Thank you, Zain.
Thank you very much. So this wraps our session for today. I learned a lot. I hope you enjoyed the talks. Thank you all for attending. And okay, so maybe the presenters can join us for one group photo.